Okay, so the next group we're going to talk about is something that we've already talked about a little bit and you kind of know a lot about, which is Helicobacter pylori. Okay, so again, these are going to be gram-negative rods. Shocking, right? And these guys will have multiple flagella. So they're able to move easily, which is good because they're going to be found in your stomach. Okay, so we already know this. They're the leading cause of peptic, which are also called stomach ulcers. And they also are a leading cause of stomach cancer. Okay, so how do they make this work? Just a little review. They're going to burrow into the stomach mucosa and cause inflammation. How do they cause this inflammation? Well, it's because they release this thing called urase. Okay? So it's possible to find a good amount of this as your normal flora. Specifically 10 to the 8 to 10 to the 9th. Okay? So how exactly does this work? So this is your urea molecule. When you release urase, it breaks down that urea into ammonia, or ammonium, and carbon dioxide. Okay? So this ammonium molecule, this neutralizes HCl in the stomach. So remember, stomach acid contains HCl. So this ammonia is going to neutralize it. When it neutralizes that HCl, you get an increase in pH. Okay? And therefore, that's what causes the stomach ulcers, because at the end of the day, your stomach is supposed to function at that pH. And when it doesn't, bad things happen. So how do we diagnose this? Well, we have what we call the urea breath test. So essentially what will happen is that a doctor will give you radioactive urea, which has got carbon-14. Carbon-14 is used for carbon dating, okay? So this is going to be radioactive urea. So the same process is going to occur if you have this. You're going to get urase, is going to break it down into ammonium plus, and instead of it just being regular carbon dioxide, it's going to be carbon-14, which is radioactive. So therefore, when you breathe back out that carbon dioxide, you're going to breathe out radioactive CO2. Okay, so that was a little bit more of a review. The next group is not going to be a review, but I've talked about it a little bit beforehand, and you watched about it in your video if you did watch the videos that I had asked you guys to watch. Okay, so it's salmonella. Okay, and salmonella's got a number of different species. There's numerous different species of salmonella. But they all have some similar characteristics that we're going to talk about here. Okay? So the first thing first is that you're always going to find them in the intestines. So they actually attach to the intestinal epithelium. Specifically, what is known as the Peyer's patches. Okay. Within those Peyer's patches, what really the Salmonella is looking for is this thing called the M cell. Okay, so this M cell looks something like this. Okay, so that's my M cell. And within the M cell, we have an endosome. So what specifically the Salmonella is looking for, they're looking for that M cell, and then they're going to invade that endosome. So this is where we're going to find my Salmonella. Okay. The other thing that they have in common, they've all got these things called pathogenicity islands, or essentially genes. Okay, the first one that we're going to talk about is going to be the invasion proteins. Most of the invasion proteins are going to be the same for all types of Salmonella. And the other thing that's going to be common among all Salmonella is going to be the toxin genes. What these genes do is they cause inflammation, cramps, fever, diarrhea, etc. So how exactly is Salmonella spread? We talked a little bit about this in the other video, but just to review, it's going to be spread through unpasteurized milk, and other dairy products. Eggs and chicken or poultry. For the most part, it's mostly going to affect humans, that are younger than 5, and older than 60 years old. Okay, so what are we affecting with those humans? Well, we're going to have 2 specific diseases that Salmonella is really responsible for. The first one is gastroenteritis. Okay, and in this case, the infectious dose for this disease is going to equal 10 to the 8 bacteria. Okay, so that means 10 to the 8 of all of the bacteria. That'll basically give us an infectious dose, okay, for gastroenteritis. Okay. Usually, you'll experience some type of symptoms within 24 to 48 hours after infection. So this is going to cause nausea, vomiting, Non-bloody stool. Remember, your bloody stool is going to be your Shiga toxin. Cramping, and a possible fever. Usually this is going to go away within a week. On the other hand, the more severe portion would be typhoid fever. Okay. Okay, and in this case, what happens here is that the salmonella will start to resist phagocytosis. By macrophages in the lymph tissue. And this will allow it for it to spread. Once it spreads, it's going to replicate in the liver, spleen, and bone marrow. Eventually, this will colonize in the gallbladder. Okay. So some symptoms of typhoid fever include you're going to have a gradual worsening fever. Usually you're going to hit the 104 degrees Fahrenheit. Okay. Headache and anorexia. You're not going to want to eat. This usually will develop within 10 to 14 days. And this will continue until you develop immunity. Okay, so those are the two major types of diseases that are caused by salmonella. And that's what you need to know about salmonella. Now the next bacteria we're going to talk about is going to be Staphylococcus aureus. And we've already talked about this a decent amount. But let's just talk a little bit more about this. So the big thing that Staph aureus causes is going to be toxic shock syndrome. Okay, and that's the big one. And the reason why we know about this is because of the super absorbent tampons. In the 1980s. So essentially what happened, women just found out about these tampons and how long they could keep them in for them. So they kept them in for a very, very long time, much longer than they should have. And essentially what started colonizing was the Staphylococcus aureus, and it became pathogenic. And what they started to experience were these symptoms. First one was a very high fever. They became hypotensive. And they got a rash. Like a very red rash. And that's called erythematosus. rash. So what happened was that 50% of those that were infected ended up making the antibodies. And lived while the rest did not and possibly died. So why did it get this way? Well, there's a couple things about Staph aureus. It does grow in high salt concentration. And since the blood that we were dealing with the super absorbent tampons contained iron, iron reacts with anything pretty much to make some type of salt. And so therefore the Staph aureus started to grow because of the reaction of the hemoglobin iron that was in there to allow for making of other salts. The reason why we already had it was so that we have it's part of the normal flora. So therefore it's considered an opportunistic pathogen. Okay? So some virulence factors. So the proteins on the surface and enzymes. So the first one, we're going to talk about protein A. So this is going to be found on the surface of Staph aureus, okay? And essentially what it does is it binds to the antibody. Backwards. Which prevents the antibodies from working. So essentially what we've got here is this is going to be our Staph aureus here. Okay. This is going to be our protein A. Essentially it makes our antibodies go like this. Okay. Where we want to bind here. Okay. So in addition, they have these elastin binding protein, collagen binding protein, fibroactin binding protein. So these three proteins are again found on the surface of Staph aureus, and they allow for the bacteria to bind to the cell. Three, we also produce this enzyme called coagulase. So this is going to block the binding of fibrin causing blood clots. In addition, they have this thing called alpha toxin. So this alpha toxin, while it doesn't work for toxic shock syndrome as much, it does work for another another disease that you saw on the Google slides. So this causes staphylococcal, scalded skin syndrome. And the last thing that can be a virulence factor is that it can be resistant to several antibiotics. So that's what you need to know about Staph aureus. The last one is going to be Strep pyrenees. Okay. And this is going to be a gram-positive. Okay, and it's going to be beta hemolytic, which means that if you put it on blood agar, it's going to get that clear area. Like I said, that we got with one of our students is when we did that together on your throat. Okay? So some virulence factors for this one. The first one was the M protein. This is on the surface. And it's going to prevent phagocytosis. And allows for adherence to epidermal and epithelial cells. This typically has a capsule of some sort. Again, this is going to prevent phagocytosis as well. It also releases streptolysin O and streptolysin S. And these are both toxins which will kill lymphocytes, natural kill lymphocytes, which is your B and T cells. Natural killer cells. And phagocytes. And the last one, unlike most other ones that cause for blood clotting, this releases a enzyme that actually prevents blood clots, which can be good and bad. It can prevent overclotting, but at the same point, if it's in a pathogenic situation, it's going to cause for you to bleed much more. So some diseases that are associated with this. One, necrotizing fasciitis, which is flesh-eating bacteria. Another one is acute rheumatic fever, and scarlet fever. And your favorite one, strep throat. So that's it. That's it for the entire semester. So hopefully you guys enjoyed having class with me. This will be all the stuff that you need to know for your exam on your final exam, which will be next week. Okay, and I'll post a little bit of an announcement of that on campus.